And we will discuss few examples of those enzymes, including DNase-1, alginate lyase, phenylalanine ammonia lyase, alpha-1 antitrypsin, and glycosidases. We are also going to discuss, inshallah ta'ala, masking of non-human epitopes so that they won't pose any immunogenic response. Also how to engineer bacteriophages and how to specifically target mitochondria for therapeutic purposes. So let's begin with DNase-1. But where we are going to use this DNase-1? We will use it for a pathological condition known as cystic fibrosis, which is a common fatal hereditary disease most prevalent among Caucasians. So this mutant CF gene is carried by 1 in 29 Europeans, whereas its frequency for African Americans is 1 in 65, whereas for Asians it is 1 in 150. So the number is slightly different for Europeans and Asians, but this doesn't mean that cystic fibrosis does not exist for Asian communities. What happens that the CF patients become highly susceptible to bacterial infections in their lungs, and that infection causes accumulation of thick mucus. That accumulation of thick mucus, as you can see in the figure here, that for the healthy airways there is no mucus, so the air flows very easily. But for a cystic fibrosis patient, not only that his own vessels become thicker, but apart from that the mucus also gets accumulated, making the airway very narrow. So the passage of air is kind of restricted, and that causes the shortness of breath in such patients. Why this thick mucus accumulates there? What is the reason behind it? So there are four elements to it. First, the infection in the bacteria, the infection in the lungs causes secretion of alginate. Second thing, the DNA which is released from the lysed bacterial cell, and these bacterial cells lysed because of the immune cells attack on it, and the immune cells which are directly related to it are the leukocytes. So they also get degenerated during that process. So those degenerated leukocytes also accumulate in the form of mucus. And the fourth thing which is present in that mucus is the filamentous actin, which came from the cytoskeleton of the damaged epithelial cells. So these four collectively make thick mucus. It clears the airways, and those enzymes are delivered to the lungs and its airways via a pump, as you can see here. This pump, inhaler or pump, delivers aerosolic mist, and that aerosolic mist contains the enzyme. You can also see the name of the drug which is marketed, and this is a BT enzyme. You can see the name Pulmozyme, which is an inhalation solution, and it is produced by the biotechnology firm Genentech. So this DNase-1 was first marketed by US biotechnology company Genentech, and what they did that they isolated the human enzyme deoxyribonuclease 1, and then they expressed this DNase-1 in Chinese hamster ovary cells in the culture. And when I said the term Chinese hamster ovary cells, that automatically indicated that a eukaryotic system was used for the expression of this protein, which indicates that there will be proper folding. There would have been proper folding of the DNase-1 protein in that culture cells. Keep in mind, this treatment is not cure for cystic fibrosis, but it do relieve the most severe symptom of the cystic fibrosis, which is the clogging of the airways in the lungs of the patients. So this drug was first approved by FDA to be used in human back in 1994, and then its sales reached to 100 million US dollars in 2000. It peaked to 600 million US dollars in 2013. You must be wondering, how could a drug for cystic fibrosis made up two blockbuster drugs? The thing is, it is not only used for relieving the mucus in cystic fibrosis patient, but it was also prescribed for chronic bronchitis, pneumonia, bronchiectasis,
emphysema, and systematic lupus erythematosus. Then there are many companies who are now marketing DNase 1. Genentech started marketing it under the name Pulmozyme, and there are many other pharmaceutical firms who are developing not only DNase 1 but also mucolytic alternatives to be used. So there was a problem which the scientists faced then, that the native form of DNase1 binds very tightly to the actin filaments within the cell. And this binding of the monomeric form of the actin actually inhibited its ability to cleave the DNA. And you can imagine how tightly it can bind to the monomeric forms of actin by looking at the inhibitor constant, which is around 1 nanomolar. So we can say, in other words, that this interaction limits the effectiveness of DNase 1 to be used as therapeutic agent for relieving the mucus which gets clogged in the airways of cystic fibrosis patient. So by then, the scientists came up with a solution on the basis of X-ray crystallographic studies of the protein. They found that the DNase 1, uh, when it binds to the DNA, there is another side of the DNase 1 which can bind to actin. So this thought of stopping the interaction of DNase 1 with the actin by targeting certain amino acids in the DNase 1 protein. And they did that by site-directed mutagenesis. For example, they changed amino acid 114 from alanine to arginine, and they also changed amino acid 65 from tyrosine to arginine. And this change of the amino acid actually led to the decrease of the binding ability of the DNase 1 to actin, and it was as low as 10,000 fold. So that was a drastic discovery of its time. And so the actin resistant mutants were synthesized, and they had 10 to 50 fold more DNase 1 activity for cutting the DNA coming from the bacteria as compared to the native enzyme. Keep in mind that it is not known whether any additional benefit might be realized by combining the amino acid changes from several actin-resistant mutants. The clinical efficacy of the DNase 1 mutant enzyme that does not bind to actin still remains to be demonstrated. All of this work which I have told you earlier, this is on the bench top, or we can say this is the lab-based results. So still its efficacy needs to be determined in the clinical setups. So with this, I am winding my talk here.